I'm Najat Khan, Chief Data Science Officer for Janssen R&D, and it's my pleasure to be with you today, along with my Janssen R&D data science team and our superb partners at MIT. We're so honored to have been named as a finalist in the prestigious Innovative Advanced Analytics Award for 2021. As I reflect over the past year and the COVID-19 pandemic, I want to share a little bit as to how our journey began. A little over a year ago, as the pandemic started exploding across many different regions and areas in the world, the scientists and data scientists and clinical teams at Janssen came together to think through how do we discover and develop a highly effective COVID-19 vaccine against moderate and severe disease to really help so many across the entire world. To do so, we leveraged technologies that we have used and invested in over the past decade, the Ad26 platform, which had been tested over thousands and thousands of participants before. And we also then leveraged data science, which was central to think through many key questions to develop a vaccine that was effective, and then also we could bring to market as quickly as possible. One of the first questions was, how do we even understand what's moderate and severe disease for COVID-19? So little was known. The second question really focused on how do we predict risk factors so that we can ensure that we're targeting those most at risk for this terrible virus. And then the third, which I'll talk more about today, is how do we predict the unpredictable? At that time, it was extremely unpredictable where and how this pandemic would evolve, what the footprint would look like. And to develop a good vaccine that's based on the number of events that you're collecting in your placebo versus your active arm, it's extremely important to be able to run the trial in areas of high incidence of COVID-19. And we have to make these decisions many months in advance. That's where the work that we have done with MIT and machine learning and predictive modeling comes in. We had to predict multiple months in advance where these areas of high incidence of COVID would be, not just at a country level, but all the way down to a county level. And I'll tell you a little bit more about what we did and the impact from this. But before that, I want to hand it over to Dr. Dimitri Bertemis.
Our goal at MIT was to develop an accurate model for COVID-19 incidence and mortality to aid governments and organizations for decision-making. The complexity of modeling this epidemic has been unprecedented due to data scarcity, governmental interventions, development of new treatments, and vaccinations. Our approach is to develop a mathematical model called Delphi, a reminder of my Greek origin, that stands for differential equations lead to predictions for hospitalizations and infections. The Delphi model belongs to the category of compartmental epidemiological models. Namely, it accounts for susceptible, infected, and recovered people. The model uses parameters that are hardwired and parameters that are estimated daily from data. The Delphi model is updated daily and makes predictions for infections, deaths, and hospitalizations. The model selects parameters so that the difference of infections confirmed versus predicted over the observed data is minimized. We solve a nonlinear, non-convex optimization problem to find these parameters. We use dual simulated annealing to achieve this objective. The model is updated daily for all 50 US states and about 140 countries of the world. My group at MIT maintains the website covidanalytics.io, but is updated daily. The model also has the capability of estimating the effect of government response, like lockdowns, restricting mass gatherings, restricting schools, or no restrictions at all. The model has three key features that separate them from others. The first has to do with underdetection of cases. The second is separating the rates for recovery and death. And finally, the model has the capability, as I mentioned, of estimating the effect of government response. More precisely, the Delphi model consists of a system of 11 nonlinear differential equations. To model underdetection, we specifically split the infectious state to include an undetected state. This allows us to estimate the true virus spread. We further separate detected patients to hospitalized and quarantine states. Finally, the rate of recovery for hospitalized and non-hospitalized patients are significantly different. Also, we have separated the recovery and deaths as separate states, and we used different rates for recovery and deaths that significantly and positively affected the accuracy of the model. Perhaps the most critical parameter in extended SIR models is the infection rate alpha. Alpha can be regarded as the average number of people each infectious person infects per day. The standard SIR model assumes a constant infection rate alpha, which does not hold true under societal intervention like social distancing, mask wearing, or stay-at-home orders. We instead apply a nonlinear correction, gamma of t we call it, to alpha, to account for the cumulative effect of government policies. Specifically, we utilize a parameterized arctan function to help us model the 4 phases of government intervention. In phase 1, as the figure suggests, it is the initial response where the population awareness is still low. In phase 2, we observe a sharp decline in infection rate as policies to control the spread go into full force and the society experiences a shock event. In phase 3, there is a saturation of the policy effect represented by the diminishing marginal returns. They observe convexity in the graph, in the decline of infection rate. And finally, in phase 4, we observe a resurgence in cases of magnitude C due to the relaxation in government restrictions and societal behaviors. The Delphi model has been part of the ensemble model of the CDC since May 2020 and continuously ever since. The graph shows the rank of the model relative to others. As you can see from the graph, the model has consistently been one of the top models for predicting infections and deaths over this time. It is fair to say that the Delphi model has performed very well, both in absolute terms, as well as in relative terms, relative to competitive models that have been proposed.
Thank you, Dimitri. I'm Jennings Xu, Director of Data Science Portfolio Management at Janssen R&D, and lead for the COVID-19 data science activities here at Janssen. Back in April and May of 2020, we realized that the truly global pandemic required a truly global approach for our vaccine trials, and that we couldn't do it alone. So little information existed at the time about how the disease would spread that we really didn't know what risk factors it relied on, how it propagated across the world, and it was clear to us that we needed to reach out to the leading minds in this field to build a model to predict how the disease would spread for our vaccine trial. That's when we reached out to Dimitri's team at MIT and collaborated closely to develop such an effort. Dimitri?
Thank you, Jennings. There were several ways that Janssen and MIT collaborated on evolving the model over the course of the pandemic. The model was initially developed at the country level and had great accuracy for certain countries, but needed to be refined for others. The model required detailed day-by-day case counts and deaths data, which the Janssen team provided from public data sources and evaluated the projections and gave continuous feedback. As the pandemic progressed, there was a need to develop models for the second and the third waves, which required deep discussions between the teams on how and where this would be applicable, as not every country, state had those dynamics.
And as Dimitri mentioned, these were very deep discussions that really led to these improvements. where we weren't satisfied, and Dimitri's team wasn't satisfied with, just being able to predict, at the high level, what the disease spread was going to look like. To really make this actionable, we had to drill down from the country to the state and even to the local county level to monitor and predict that spread of disease. Each country, state, or county had to be modeled, evaluated for data completeness and accuracy, and refined individually. We went a step further than just utilizing the outputs from the Delphi model by itself. Because for Janssen, the critical thing was to take those outputs and really utilize them to be effective in selecting our sites and activating the right sites for our trial. So to that end, we also applied additional quantitative and qualitative inputs to inform our final site selection and activation process, as this would give us what we needed to run a global, diverse trial. This was a combination of descriptive analytics with the predictive outputs from the Delphi model, and includes some of the aspects that you see below. For example, age distribution. We had to be certain that we would be able to cover the elderly population in our trial. And so we actively looked at where our sites were and would they be able to recruit from the elderly population. Racial and ethnic distribution. We needed to make sure that this was a fully, truly diverse trial. And so from day one, we were looking at how could we improve our diversity and be in the right spots and have the right outreach to achieve that diversity. Policy enforcement. We needed a variety of areas that had lockdowns or opening up, school closures or not. And so we looked at sites in a variety of different areas. Population metrics, socioeconomic factors, and social compliance were additional factors that we looked at, that we pulled information from on a daily basis to help guide those decisions.
We collaborated very extensively. The team met multiple times a week and even multiple times a day during critical periods in order to discuss, refine, and evolve our outputs to deliver the most impact for the vaccine program.
Absolutely. It was a truly collaborative team effort. Every single person you see on the screen here and many others who couldn't fit onto this Zoom screen, we all had to work together, as Dimitri mentioned, multiple times each week, every day, to deliver the output and really help with our vaccine program. To discuss that output further, I'll turn it back to Najat.
Thank you, Jennings and Dimitri. It's great to see the collaboration and the innovative predictive models and the design that was developed. So, what was the outcome? I'll walk you through a few of the different outcomes and how they were fundamental to improving our design and execution of the trial. So first, by going in an unbiased approach, just following the data, following where the predictions were leading us, it resulted in a truly global trial across 3 different continents, the US, LATAM, as well as Africa. Let's dive into this a little bit more. So in terms of the predictions and the accuracy, the machine learning models that we developed had about 90% accuracy, not at a country level, but at a county and state level, and consistently across all of the different countries. So you can see the countries that we went to. This was extremely important for us because we ended up getting really, really rich data from participants across all of these different regions. We really appreciate the participants who joined our trial, and therefore the analysis truly reflects our participants across all of these different regions. And the 90% accuracy, if we drill in a little bit more, it really comes to life what that truly means. So if you look at this slide of the US, the first build that you're seeing here is the counties that were predicted to have high incidence, above a threshold that we wanted for the trial. Now, as you see the second overlay, all of the different sites that we actually ended up picking, as you can see, mirror quite well the predictions because we really were data-driven in terms of where we put our sites. And then now, if you see the third overlay, you see the reality. So this was predictions, but in terms of what was real, where were there areas of high incidence? And there's really high congruence and convergence of that between the predictions and what happened in reality. The other thing I'd also say is that it helped us by ensuring our predictions and our design and our models incorporated racial elements, incorporated socioeconomic elements. We ended up with one of the most diverse COVID-19 trials to date, both for Latinx and also for African Americans. If you double click a little bit more, you'll see even in specific states, the regions where we had high incidence that we predicted, where we had our sites, and where there was really, in actuality, high incidence as well. This kind of precision was critical to us in terms of running a trial as efficiently and effectively as possible. And we didn't just do this for the US, we also did it for ex-US. So let's look at Brazil and South Africa. Very high accuracy in terms of the predictions across all of these different regions. Challenging environment, right? Because a lot of the data sets were not available. There was missing data. Very different data quality versus what we had in the US. But the team worked diligently in order to overcome that, using a lot of the methodologies that they described. And also, I would say, back testing. The amount of back testing we did to ensure we optimized our models was critical to our success. The other thing I'll say is by honing in on areas where we predicted there'd be high incidence, we also ended up going to areas where there are high variance, which there is biologically hypothesis-driven, it makes sense. But I think this is a fundamental important point for us to remember, is by having clinical data, really robust clinical data on protection and efficacy across both variants in South Africa and Brazil, we really truly were able to develop a vaccine that's relevant for our pandemic and the nature of the pandemic today. And so in summary, predictions are great, accuracy being high, we all want that technically, but it's really what outcomes they drive. For us, it was having a highly rich data set where we could do a lot of subgroup analysis. This is critical to really understand the efficacy of the vaccines and the safety in as many people, in as many types of groups as possible. We accelerated timelines. How much? About 6 to 8 weeks. Every day counts in a pandemic, so this was a huge win for us. It also helped reduce our sample size, our trial size, from 60 to 45,000. Why is that important? By ensuring that we have a small sample size, but diverse and reflective of those that we would vaccinate in the real world, we were able to accelerate our timelines. And this was, again, resulted because of the fact that we had really rich data. And, I want to underscore the last point in terms of diversity. We took this really, really seriously. And we do that not just for our COVID-19 trials, but for all trials across Janssen. When I take all of this in and the fact that we had great data in variants, and the outcome today of people being vaccinated across the globe with Janssen's vaccine, this is how the advances in AI, machine learning, can really solve problems that matter not just to a company, but to the entire world. So thank you.
Thank you, Dimitri. Thank you, Najat. Thank you to everyone for allowing me to say a few words. We work together to deliver a vaccine that's a definitive solution for one of the most important healthcare crises of the last century. And we did that together through use of modeling and simulation, the use of data science. This was a close collaboration between the Janssen Data Science Group with Najat and Jennings, and with the group at MIT with Dimitri. And what did we do here? We created a model that we compared to a number of other alternatives, and it turned out to be the best. It was the best and able to predict the course, the ups and downs of this COVID-19 pandemic. Why did we need to do that? We wanted to maximize the incidence that we would see in the trial by being very choiceful in where we went, what country, what province or state within a country, what place within that province or state. And we were able to do that successfully. Why did we want to do that? For two reasons. One, we wanted a diverse population. And that was because we want the population that we study in phase 3 in any therapeutic or vaccine, including this one, to look like the population we're going to treat or vaccinate. And we were able to achieve that. We had the most diverse population in the United States of the trials that were conducted, thanks to our efforts here together. Why else did we want to do that? We want to maximize the incidence of COVID-19 during our trial. And why would we want to do that? Because the more the incidence is in that region, the more likely the correlation to a higher number of cases we would have in the trial. And the greater the number of cases, the more quickly and the more easily we are able to distinguish a vaccinated person from someone that was given placebo. And that's exactly how it played out. A really important bonus feature of having high incidence is that we also saw the emergence of variants. When you have high incidence, it implies that there's a lot of virus replicating. And when virus replicates, there's a chance each time that it mutates. And the more virus replication there is, the greater chance that problematic variants emerge. And that's exactly what happened in Brazil, where the P2 variant emerged during our trial, and in South Africa, where B1351 emerged during our trial. So all in all, we did it. We did it together. We did it in close collaboration with Janssen's data science team and Dimitri's team at MIT. And we, in fact, delivered on a definitive solution for this pandemic. Thank you very much.